Just as there's an ivory trade for land mammals, there's one for marine mammals as well. The bones of these marine mammals are called mermaid ivory. Most of this trade is focused around the bones of the now extinct Steller sea cow. The bones are used for many purposes, including being fashioned to knife and tool handles and decorative pieces. While the trade of Steller sea cow bone is legal because of their extinct status, the trade of other bones of other animals is not. Bones of gray whales, white-beaked dolphins, and other species are routinely passed off as bones from Steller's sea cow. The biggest question facing regulatory agencies is how can one tell the difference between legal and illegal mermaid ivory in a timely and efficient manner? If such a method were to exist, it would boost efforts to crack down on the sale of illegal bones. George Mason researchers Lorelei D. Crerar, Elizabeth W. Freeman, E. Christian Michael Parsons, and Howard researcher Darrell P. Domning believe they have found just that. With their research paper, A Legal Trade of Marine Mammals Bone Exposed, Simple Test Identifies Bones of Mermaid Ivory or Steller Sea Cow, they believe they've solved the problem with polymerase chain reaction amplification and electrophoresis. They begin their process by collecting bone dust samples from the bones in question. They then take this dust and extract the DNA from it. They then take the pellets of DNA they had extracted and run them through polymerase chain reaction. Polymerase chain reaction is a method used to turn a small amount of target DNA into a large amount of that same target DNA. To start, the processed DNA you want to amplify is added to the mixture along with loose nucleic acids and the DNA primers. The DNA strands are then broken in two using heat. Upon cooling down, the primers bond to the specific point on the strand that they fit on, and they start making complementary strands of DNA using loose nucleic acids. The mixtures are then heated up again and the strands are again separated. As the mixture cools down, the primers bond and again make complementary strands of the DNA. The process continues in this way, doubling the amount of DNA each time. Over the course of hours, it is possible to turn several strands of DNA into several 100 million or more. The researchers take their newly acquired DNA samples and run them through the next step of electrophoresis. Gel electrophoresis is an easy way to distinguish DNA samples from each other. First, an agarose gel block with wells in it is submerged in a buffer solution. Then, a negatively charged terminal, or cathode, is placed next to the wells on one side of the block, while a positively charged terminal, or anode, is placed on the other end of the block. The DNA of the samples are inserted by pipette into several of the wells. The other two wells are filled with a positive control, like DNA from a Steller sea cow, and a negative control, like DNA from a gray whale. The DNA stays in the wells because it is denser than the buffer solution that surrounds it. A current is then run through the solution and the block. Since DNA is negatively charged, it will be pushed away from the cathode end and will be attracted towards the anode end. The distance the DNA travels through the block is determined by its mass, as it's easier for the smaller pieces of DNA to move through the block. When the current is stopped and you examine the block under UV light, the DNA in the block will glow. The DNA will spread out into distinct bands in the block. Each species has a distinct DNA spread through the agarose block. By quickly checking the block, we can see which samples are in fact Steller sea cow bone and which aren't. The samples which are not Steller sea cow can then have their DNA sequenced to discover which animal it actually is. When used, this method proved to be highly effective at determining whether a sample was in fact sea cow or not. However, one problem with this method is that there are very few sequences of DNA available from GenBank that are useful for identifying samples. If these sequences were to be added, this method would be proved to be highly effective at ending the illegal ivory trade.